All right, so Krebs steps and Krebs cycle. There are a lot of names of molecules here that we are not going to worry about. I'm going to write the names of the molecules down when I draw a molecule, but I'm going to put them in parentheses if you don't need to know them. If you do need to know them, I won't put it in parentheses, but there are very few of those. Okay, so we are concerned in lower level biology classes in bio honors. You do something that's called carbon accounting. Carbon accounting is where you just look at the number of carbons in each molecule. We're still going to do that. It's still important. But we're also going to add another level to this, which we are going to do something that's called electron accounting. And electron accounting turns out to be the much more important thing because of the fact that we want to capture those electrons for use in NADH and FADH2, right? Yes? Let's get out of my Amazon shopping cart and back to where we're supposed to be. All right, so let's talk about the prep steps. Sometimes the prep steps are called the formation of acetyl-CoA or acetyl-coenzyme A. These are two ways of referring to this stage. And so in this stage, you're going to need two things. The first thing that you're going to start with is a 3-carbon chain. This 3-carbon chain is called pyruvate. You should recognize the name pyruvate because pyruvate was the product of glycolysis. And how many pyruvates did we produce in glycolysis? That means that we are going to go through this stage 2 times. Okay? So pyruvate is going to combine with coenzyme A, which we are going to abbreviate CoA like this. Coenzyme A is a large molecule. It's a large protein. Coenzyme A. And it's a coenzyme. In this case, coenzymes are allowed to be consumed in a reaction, but this one happens to not be consumed. Right? So coenzyme A is going to be what's called a spectator. It's going to shuttle some molecules back and forth. It's going to participate in 2 reactions, but since it's both a product and a reaction, or a product and a reactant, we would say that it's a spectator in this case, and so we wouldn't worry about it. Come on, I wanted that to be a black line. All right, black line here, pyruvate, something like this. All right, coenzyme A is going to come into here like this. It's going to join in, right? And as a result of that, we are going to remove a carbon dioxide. Okay? Now, this carbon dioxide has double bonds. Anytime we go from having single bonds over here to having double bonds, we are going to free up electrons, right? That's sort of what a double bond means, is you're going to be able to gain electrons from it because you have less electrons that are participating in the bonds because they're sharing more electrons, and so you can free up electrons to be used for other things. Now, what are these electrons going to be used for? They are going to be used to take an NAD plus, right? And this NAD plus is going to form NADH. Why does it do that? It's going to gain 2 electrons, gains 1 electron and becomes NAD, gains another electron, becomes NAD minus, and then it picks up the hydrogen ion to become NADH, right? Okay. So now we've lost a CO2, so now we've done our electron accounting, we've got to do our carbon accounting. So a carbon leaves here as CO2. That means that we only have a 2-carbon chain. It looks like this. This 2-carbon chain bonds to coenzyme A. And that 2-carbon chain is called an acetyl group. Okay, so that makes this molecule that forms over here acetyl-coenzyme A. Acetyl-CoA. Right? So the prep steps produced an acetyl-coenzyme A, which is why it's sometimes called the formation of acetyl-coenzyme A, because that's what's happening here. Okay, so let's look at the inputs and the outputs for the prep steps, or the products, or the reactants and the products. And this is the reactants and the products specifically for the prep steps. So we're going to go through these twice, and so that means that we are going to require 2 pyruvate. We're also going to require 2 coenzyme A. We're going to need 2 NAD pluses. And then our outputs are going to be 2 acetyl-CoAs, 2 CO2s, because those CO2 left as a waste product, right? And then 2 NADHs. All right, so let's talk about where all these things are going. The acetyl-coenzyme A is used in the next step, which is the Krebs cycle. The CO2, what's the CO2? Was it used for anything? No, so it's waste product. NADH, what's it carrying? High energy electrons, and therefore we want to use it where? In the electron transport chain. Okay? So then we're going to go into the Krebs cycle, which is sometimes called the citric acid cycle. And the citric acid cycle is going to start with that acetyl-coenzyme A. Which looks like this. Okay, you got 2 carbons on this coenzyme A. And it's going to join up with a 4-carbon chain. This 4-carbon chain is called oxaloacetate. Or sometimes oxaloacetic acid, because any of these things are going to be able to mix with water and become an acid. And so this oxaloacetate is going to join up here with 2 carbons from acetyl-coenzyme A. All right. The acetyl-coenzyme A is going to drop off its 2 carbons, and it's going to leave as coenzyme A. So no longer is it acetyl-coenzyme A, now it's just CoA, right? So because the CoA was used up here and then produced down here, it's just going to cycle back and forth between these 2 things, right? So now we've got 1, 2, 3, 4 carbons, 5, 6 carbons, right? So these are now a 6-carbon chain. This 6-carbon chain is called citrate. Citrate mixes with water to become citric acid. So notice how this one's not in parentheses. Why do you think it's important to know that the first carbon, or the first 6-carbon chain here is citrate or citric acid? Because it's the citric acid cycle. That's why we call it the citric acid cycle, because that is the first thing that gets produced, right, is citrate. All right, so now each time we go along here, what we're going to do is we are going to remove a carbon from this chain and turn it into carbon dioxide. That's going from a single bond to a double bond. And every time we do that, we are going to be able to take an NAD plus and turn it into an NADH. Okay, so we remove a carbon. That means now instead of 6 carbons, we've got 5 carbons. 2, 3, 4, 5 carbons. This molecule called alpha ketoglutarate, parentheses, of course, because you don't need to know that one. Alpha ketoglutarate is going to lose a carbon from carbon dioxide here. I am skipping some steps, or not necessarily skipping, but combining some steps so that they are more similar. So each step is more similar, where you have a CO2 leaving, and then you are getting a high energy or 2 high energy electrons from it. But that's fine because we're just doing, like I said, carbon accounting and electron accounting. So we lost carbon here, so now we're at 4 carbons. 4-carbon chain that's called succinate. Succinate breaks into fumarate. We're not going to show that step. We're just going to go way over here. This is going to be a big step. We're going to do 2 steps at once here. The first step is succinate into fumarate, right? And then the next step is fumarate into malate, right? So we're just going to say that this is malate. And then malate into oxaloacetate. Okay, but I wanted to show you guys what happens here with succinate into fumarate, actually, and it looks like this, right? So here's the structural diagram of fumarate and malate, and I just want to we're just going to do electron accounting, right? So let's electron account. Every time you see a line, that corresponds to a pair of electrons, right? So this is 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26 electrons, right? Over here, we're changing from succinate to fumarate. 2, 4, 6, 8, 10, 12, 14, 16, 18, 22, 20, 22, 24. Right? Yes? So we went from 26 electrons to 24 electrons. How did we do that? Because we knocked off this hydrogen and this hydrogen, right? So now we are going to change this bond here into a double bond in the middle, and now we've got these 2 extra electrons over here, right? So if we have 2 extra electrons, those 2 extra electrons can join up with FAD. It becomes FAD minus 1 and then FAD minus 2. So then now it's FAD minus 2, so it's going to pick up 2 H plus ions from the surrounding area and become FADH2, which is a high energy electron carrier. Every time we show one of these transitions between like NAD plus and NADH, what we're showing is that 2 high energy electrons, or 2 electrons, have been removed from one of those molecular compounds. Now before, we also were showing the CO2s, so it was easier to say like, oh yeah, there was something changing within that molecule. But when you go from a 4-carbon chain to a 4-carbon chain, right? So this is still 4 carbons, 1, 2, 3, 4, and this is 4 carbons, 1, 2, 3, 4. But we just added a double bond in order to get those electrons out, right? So in this switch here, succinate to fumarate, we are going to take an and we're going to make an FADH2. And then between fumarate and malate, there's going to be a little bit of magic that occurs where a coenzyme A comes in and helps again and produces a molecule that's called GTP. In order to produce GTP, you need GDP. Not quite. Close. All right. Now, we've never heard of GTP before. We haven't talked about it. But it's the thing that's happening here. So why GTP? What does it mean? What is ATP? What does A mean in ATP? Adenosine, which is a nucleotide. It's a nucleoside, right? So it's the nucleotide adenine, and then we remove the phosphate group. So this is the guanine nucleotide. So this is guanosine triphosphate, right? It's very similar to ATP. In fact, for all intents and purposes of this class, we can consider it the exact same as ATP. Right? But it just happens to be that the enzyme that produces it here produces GTP rather than ATP, right? And that's a pretty common thing in your body. In fact, GTP is really common for muscle contractions. It's used even more than ATP is for muscle contractions. But that's neither here nor there. All right. So then we have one last step, and that is from malate to oxaloacetate. And going from malate to oxaloacetate, we're going to gain one more double bond. And when we gain one more double bond, we are going to then produce an NADH. Okay?
And it's not NAD plus NADH, just NADH.
Oh, sorry. Yeah. Sorry, I forgot I'd been doing that. NAD plus to NADH. Yeah, otherwise, if it wasn't that, instead of gaining 2 electrons, you'd have to gain the whole nicotinamine adenine dinucleotide, which would have to be incorporated into the malate, and we don't want to do that. All right, so NAD plus turns into NADH. All right, so this is the citric acid cycle, or the Krebs cycle. Right? The purpose of the citric acid cycle is to capture high energy electrons. Right? How many times can we go through the citric acid cycle per glucose? 2 times, right? So 2 pyruvates are produced in glycolysis, 2 pyruvates go into the prep steps, 2 acetyl-coenzyme A's are produced, 1 acetyl-coenzyme A per cycle in the Krebs cycle, right? And you'll see here that this final step from malate to oxaloacetate reproduces the oxaloacetate that will then pick up the acetyl group here to become citrate again. So it's a cycle. Right? So just like the Calvin cycle begins and ends with RUBP, the citric acid cycle begins and ends with oxaloacetate. Oxaloacetic acid, but that's okay. Oxaloacetate. Questions on this? Let's talk about reactants and products. Two acetyl-coenzyme A's. Yes. Yeah. This is reactants and products per glucose molecule. All right, so 2 acetyl-coenzyme A's. So we are going to need 6 because we're going to produce 3 per cycle. So we'll need 6 total NAD pluses. 2 GDPs, guanosine diphosphates, 2 FADs. The acetyl-coenzyme A's get their acetyl group removed from them and become 2 CoAs, coenzyme A. The 6 NAD pluses gain 2 high energy electrons each to become NADH. The two GDPs become GTPs, which we can think of like ATP, right? And the two FADs become FADH2s. All right, so just like we have been doing here, where are the coenzyme A's going to go? Back to the prep steps where they're used. Where are the NADHs? Yes, they're going to go to the electron transport chain because they're carrying high energy electrons. What about the GTP? Cellular processes, ready for use. It can be used for whatever now. They're good to go. FADH2? Electron transport chain because they're carrying high energy electrons. So we've gone through the vast majority of the process of cellular respiration, and we've produced a whopping 4 ATP equivalent, right? So we produced 2 of them in glycolysis, and we produced 2 of them in Krebs cycle, right? And we know that aerobic cellular respiration produces 36 to 38 of them, right? So these 4 that we've talked about are through what's called substrate level phosphorylation, meaning that they are part of an enzymatic response, right? But that means that there has to be 32 to 34 of them that still haven't been made, right? So 32 to 34, that's going to be made in the electron transport chain. How are we going to make them? With what energy? Well, we've produced a bunch of stuff, right? We've produced 6 NADH here. We produced 2 NADH in the prep steps, and we produced 2 NADH in the process of glycolysis. And so that was a total of 10 NADH that'll get added in. So 10 NADH times 2 high energy electrons each. That's 20 high energy electrons, right? Plus the 4 high energy electrons, slightly lower energy, but they're still high energy, electrons from the FADH2, right? And so that's a lot of high energy electrons that can be used to pump H+ ions and then go through oxidative phosphorylation to produce ATP, right? And that's where the vast majority of this is going to happen. FADH2, so this process here, there's never going to be a question on the AP exam. Mark my words. Never will there be a question where it says, draw the citric acid cycle, right? Or not even draw the transformation from alpha ketoglutarate to succinate, right? Not a thing. Never going to happen. Okay, but what it might say is, how is NADH produced from NAD+ in the citric acid cycle, right? In which case, you need to know that the whole way that you can get NADH is by taking electrons away from these large molecules, right? So if it showed alpha ketoglutarate, if it showed this thing right here, right? This part, alpha ketoglutarate, this 5-carbon chain turning into a 4-carbon chain, and it showed the CO2 coming off and the NAD+ turning into NADH, and it said explain how NADH is made here or why NADH is made. Well, you would need to know, well, the reason that NADH can be made here is because we are going to take 2 high energy electrons from alpha ketoglutarate away, and we're going to replace them with a double bond in carbon dioxide. Right? So the 2 high energy electrons are added to NAD+. Again, it becomes NAD, then NAD-, and then that turns into NADH, right? Or a good question would be, why is FADH2 made in the succinate to fumarate transformation instead of making another NADH? What do we know about FADH2 versus NADH? It's slightly lower energy, right? So what do we know about the electrons that are coming off of succinate going to fumarate? They're slightly lower energy. So why can't you make an NADH from that transformation? They're not high enough energy electrons that we're removing, right? So whatever their bond energy was, whatever orbitals they were in, energy levels they were in, were not high enough to be able to go into the NADH. they were only high enough to be able to go into the FADH2. Make sense? Yeah.
So I'm a little confused. Coenzyme A takes 2 carbon molecules from pyruvate.
You can think of it as a shuttle. It's shuttling 2 carbons from pyruvate onto oxaloacetate to make citric acid.
And then it becomes free after the molecule.
Yep, it's just passing them along, right? So Acetyl-CoA passes its 2 carbons over, and then it's just CoA where it leaves.
And we're only releasing 2 CO2 in this whole process?
Yes. Yeah. Yeah.
Does the CO2 molecule in any way affect the or does it just get released?
Is the 2 CO2 what?
Like does the CO2 like contribute to like the oxidation of NADH or not?
Yes. CO2 has a double bond. It's two carbon or 2 oxygens double bonded to a carbon, and so because the CO2 itself has those double bonds, that's where those electrons are coming from. That's the molecule that has less electrons than it did before. Right? I guess I didn't put that in. Yeah, we should put that. We should put the CO2s in here. Thank you. Yeah, so it's 2 per cycle. Right? Which is waste. Whoa. And did we list them up top too? Yeah. Okay, so so now we've accounted for all of our CO2s, right? So 2 CO2s are given off as products in the Krebs cycle, and 2 of them in the prep steps. And when we look at the overall equation, how many CO2s are in the products? 6, right? C6H12O6 plus 6O2 yields 6 CO2 plus 6 H2O, right? So 2 of them here, 4 of them here. So what can we be pretty sure isn't going to happen in the electron transport chain? We won't have carbon waste in the electron transport chain. But what will we produce? Water, right? H2O, which is the other thing, because we haven't accounted for water yet. Questions? All right, that is it.